Welcome to Educon. Coming back to cell unit of life. Now we'll be continuing with the prokaryotic cells, 8.4 section from class 11 NCERT biology test. Okay. So prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. So it means, so what are these prokaryotic cells? They include bacteria, blue-green algae, mycoplasma, and pneumonia-like organisms. And they are smaller. Smaller means they are smaller in size, and multiply more rapidly means reproduction, or giving rise to new cells. It occurs more rapidly than the eukaryotic cells. They may vary greatly in shape and size. मतलब क्या है? Maybe the shapes are different and the sizes also varies according from different species to species. The four basic shapes of bacteria are Bacillus rod-like, Coccus spherical, Vibrio comma shape, and Spirillum, which is spiral in shape. The organization of prokaryotic cells is fundamentally similar, even though prokaryotic cells exhibit a wide variety of shapes and function. See here, what is the meaning of this? The organization of prokaryotic means different cells. They are fundamentally similar. Means fundamentally they are similar, even though prokaryotes exhibit a wide variety of shapes and functions. Coming next, all prokaryotes have a cell wall surrounding the cell membrane, except mycoplasma. You should remember this one. Okay. So the question will be, cell wall is absent in animal cells, mycoplasma, and some other two options, all above, like that. Otherwise, under how we can make question is, which of the following microorganism doesn't possess cell? So here the options will be, you'll be having the option including mycoplasma. Okay, the fluid matrix filling the cell is the cytoplasm, as I told you before when we had a brief introduction about cell, that is the overview of cell in previous section. That cytoplasm is the place where most of the cellular activities takes place, which keeps the cell alive. Okay, there is no well-defined nucleus. So already we discussed about this. Prokaryotes, they don't have any kind of membrane surrounding their nucleus where their genetic material is exposed or naked, which is not enveloped by any kind of membrane. In addition to genomic DNA, apart from the main DNA, many bacteria have small circular DNA outside the genomic DNA. See here, the genomic DNA will be a single chromosome or circular DNA. Remember this, okay? In bacteria, what is the genome? It is a single chromosome or circular DNA. Okay? Many bacteria have a smaller circular DNA outside the genomic DNA. Means it is not associated with the main chromosome or DNA. So they are present outside. These smaller DNA are called plasmids. This is also important question. Plasmid DNA confers certain unique phenotypic characters to bacteria. Okay, what is such characters? Resistance. What is the phenotypic character here exhibited by the bacteria? Is resistance to antibiotic. So for example, before we used to take, when we got any kind of infection and we started taking medicine, sometimes we are cured, sometimes we are not, because the resistance to antibiotics or the drug occurs in our body. So most of the pathogens or the bacteria, they build such a certain kind of resistance from the drugs we are taking. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with the foreign DNA. That we'll discuss in the later chapters. Okay. Nuclear membrane is found in eukaryotes. Yes, that already we discussed in overview of a cell. And also no organelles like the ones in eukaryotes are found in prokaryotic cell except for ribosomes. Yes, because ribosomes, we discussed that ribosomes are present in both prokaryotic cell and also eukaryotic cell. Okay, before we discussed in overview of a cell. Okay, and prokaryotes have something unique in the form of inclusions. Apart from ribosome, prokaryotic cell, they do have certain kind of certain unique structures called inclusion, which is formed by the infolding of the, here you can see here, infolding of the plasma membrane. So a specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infoldings of cell membrane. So the membrane folds, and by the membrane infold itself, okay, forming mesosome, and also it increases the surface area of the plasma membrane. Now we'll go for the next section in another video. Okay, we conclude with this prokaryotic cells. Brief introduction: what are prokaryotic cells? What it includes? What we study? It includes bacteria. Okay, bacteria, mycoplasma, pneumonia-like organisms, and blue-green algae. And also they are smaller and multiply rapidly, and they occur. Their size are different, and also sizes. They may vary greatly in shape and also size. Different types. What are the basic shapes of bacteria in prokaryotes? Rod-like, spherical, comma, and spiral. Okay, and the organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. Okay. So with this, we conclude this section, and we'll move into next section. And once you finish listening to my lecture, kindly read the textbook again and again, because it will make you remember the things in proper manner. And on the left side, we are seeing a complete a diagram showing comparison of eukaryotic cell with other organisms. Okay, here PPLO and typical bacteria is 1 to 2 millimicron. Pneumonia-like organisms about 0.1 millimicron in size. Here typical bacteria is 1 to 2 micron. Remember, viruses 0.02 to 0.2 millimicron. So with this, we conclude this section. We'll move on to the next section, that is cell envelope and its modification. Thank you.